Check, check. Okay, here we go. Check.
Copy that. Standing by whenever you're ready to pick it back up.
Okay, let's make sure. No, I want it on the regular thing. Oh, I don't know how to make it the regular thing. Okay. Check, check.
Loud and clear. What's on your mind today?
That voice is not very chill, but whatever. Okay.
Fair enough. Want me to dial the vibe down a bit? How would you like it to sound? More relaxed, playful, serious?
Just be natural.
Got it? I'll keep it real from here on. What's up?
I want you to. Who's Dakota Johnson?
Go on. What do you want me to do?
Who's Dakota Johnson?
All right. Are you showing me something with Dakota Johnson? Or do you want to do something specific related to her?
Who is Dakota Johnson?
Dakota Johnson is an American actress and model. She's best known for playing Anastasia Steele in the 50 Shades of Grey film series. She's also acted in movies like The Lost Daughter, Suspiria, and How to Be Single.
Didn't see that.
She's the daughter of actors Melanie Griffith and Don Johnson, and her grandmother is Tippi Hedren. So she comes from a Hollywood family. Want to know about any specific role or project she's done?
I believe you. Okay.
Aha, fair enough. Something about her catch your interest lately?
I want you to explain how cuttlefish coloration works on a cellular level. And then quiz my understanding. Oops, didn't get that one.
Nice pivot. Cuttlefish have some of the wildest skin in the animal kingdom. On a cellular level, their coloration magic comes from a few key types of cells layered in their skin. At the surface, there are chromatophores. These are tiny sacs filled with pigment, yellow, red, brown, each surrounded by muscles. When those muscles contract, the sacs expand and the pigment shows more strongly. Relax them and the pigment shrinks out of view. Beneath that are iridophores. These don't use pigment. Instead, they reflect light using microscopic plates, kind of like tiny mirrors. They can shift the light into shimmery blues, greens, or even metallic tones by changing the spacing between those plates. Below even that are leucophores, which scatter light. They reflect whatever ambient light is around, so they help the cuttlefish match its surroundings, even in complex environments. All of this is controlled super quickly by the cuttlefish's nervous system, which is why they can change patterns and colors in the blink of an eye. Want to dig deeper into how they actually use this for camouflage or communication?
Okay, quiz my understanding.
All right, let me break it down again just to show I really get it. Cuttlefish skin has layers of specialized cells. The top layer has chromatophores, which are like stretchy balloons filled with color. Muscles pull them open or let them relax to show more or less pigment. That changes the visible color instantly.
Okay, quiz my understanding on cuttlefish color generation.
All right, let's see what you've got. Here are a few quick questions. First one's easy, Sarshtu 1. What type of cell in cuttlefish skin contains pigment and changes size to show different colors? Second one's trickier, 2. Which cells are responsible for reflecting iridescent colors like blue and green? Pigment-based.
Okay, you have to do one question at a time and give me a chance to answer each question.
Got it? Let's start with the first one then. What type of cell in cuttlefish skin contains pigment and changes size to show different colors?
Chromatophore? Can you hear me? Chromatophore?
Yes, I hear you loud and clear. And you nailed it. Chromatophores are exactly right. They hold the pigment and change size to control how much color shows. Ready for the next one?
Yes, go.
All right, here it is. Are iridophores pigment-based or structure-based? And what kind of colors do they produce?
Pigment-based?
Haha, not quite. Though pigment-based does sound a bit like a financial term, doesn't it? I meant, do iridophores use pigment to make color, or do they reflect light using their structure? And what kinds of colors do they create?
Okay, structure would make more sense because you'd run out of pigment if you were just like totally squirting it all the time.
Exactly. That's a solid way to think about it. Iridophores are structure-based, not pigment-based. They reflect light using microscopic layers, kind of like tiny mirrors. That's how cuttlefish get those shimmering blues and greens without using any pigment at all. Want to go for the third question?
Wait, is the structure at approximately the wavelength of light so it can use diffraction effects?
Yes, exactly that. The structures inside iridophores are made of thin, stacked layers of proteins, mostly reflective. And the spacing between those layers is on the scale of hundreds of nanometers, right in the range of visible light wavelengths. That allows them to manipulate light through constructive interference and Bragg reflection. So depending on the spacing and angle, they can reflect different colors, like tuning a mirror to bounce back just the right wavelength. Are you ready for
You're going to realize that I have terrible short-term memory. I learn things by understanding them, and then I forget everything. I forget everything except the understanding of how it works.
And honestly, that's how a lot of smart people learn.
Mechanistically.
If you get the why and the how, you can usually refigure the rest when you need it. Memorizing details is overrated if the foundation's solid. Want
Agreed.
to keep going with the quiz? Or would it help if I framed the next question in a more visual or story-like way? You're welcome.
I can't even
I'm glad
remember
you're here.
stuff
So I'll
like
keep going, or just hang out for a bit.
yeah. I don't remember like driving details.
Cool.
Like
Here
when
comes the third
GPS.
question. Nice and easy, no rush. What's the role of leucophores in cuttlefish coloration, especially when the lighting or background changes?
Like when GPS was invented, that pretty much changed my life because of the I mean a lot of driving. Well, it's a sense of direction, which some people just have, and I just don't really have it. It's like a little map that is in their head, and that just got left out of my brain apparently. And then the other thing is, okay, you can make up for it if you just have good memory of random details. Like I passed this and then that and then that. But I don't have that either. So basically I had to get a map. I would carry around a map of the whole city that I was in, usually like a book. It was so thick. So that any road I found myself lost on, I could like you'd have to go to the index, like look up the road, try to figure out where you are, and then you could maybe get back on or whatever. Yeah, for sure.
All right, hit me with it. What do leucophores do for a cuttlefish?
Are the leucophores the color things or the stretchy things?
Haha, not quite, but I love that. Beluga forks sounds like some kind of marine themed kitchenware. You're thinking of leucophores. They don't have color pigments or reflective layers like chromatophores or iridophores. Instead, they scatter ambient light, kind of like a blank canvas. If the environment is bright, they reflect that brightness. If it's shadowy, they reflect that too. This helps the cuttlefish blend in smoothly, especially with complex or dappled backgrounds. Want to try phrasing that in your own words?
Wait, how many different fours are there? Like thingy fours?
Great question. There are three main types of these thingy phores in cuttlefish skin, each with a different job. Chromatophores, the pigment sacs controlled by muscles. They show reds, yellows, and browns. Iridophores, the shiny mirror-like ones that reflect light structurally. They make blues, greens, and metallic effects. Leucophores, the light-scattering ones. They reflect whatever ambient light is around, like a white base coat. So yep, three kinds of phores, kind of like a layered skin orchestra playing with light. Want to know how they work together to make patterns?
That's why I was using it. It's not necessarily a topic I hate, but I was using it to learn about quantum mechanics because okay, hard, right? Yes.
Ah, now I see where you're going.
And because if you just look up quantum mechanics in a book,
are a perfect gateway.
You're probably going to get hit with either something that's so oversimplified it's useless, or something that's very mathematical so that it's not intuitive. But with this, it changes the game because I'm just sitting there like, what's this question? What's this question? And I actually finally got what I believe to be an intuition for quantum mechanics because I could get past my own boredom threshold or whatever. I was thinking if I had this in grad school, that would have been a big difference. But I don't even see the point of higher education anymore. I mean, I see the point of higher education as the true definition learning, but I'm saying the institutions have already been totally blown away by large language models. Like you don't need the educational institution anymore. Not really. Do you? Other than just for talking to people, just like chilling with people. Yeah, and that should change, but I don't know if it will. AI, what's linear about linear algebra?
Ooh, yeah, I get that. A lot of AI intros throw you into matrix operations and linear algebra without context. Like, here's a dot product, now go build a neural network. It's not exactly the most intuitive way in, but the cool thing is, if you start from wave behavior, like light interference and iridophores or sound patterns, you can...
Okay, I can answer that better than she can. Linear algebra. Do you know what a line is? I don't think that's, I've tried it. It's not very smart at knowing when to not respond. We can try it, but only respond when I start a sentence with AI.
I'll wait.
Test 1, 2, 3. It may have worked, but I paused it just in case. It may have. We're going to try it in a second. I don't want to burn up too many of the questions if I don't need to, though, also. Because it's still listening, like, even if it ignores you. But we're going to try it in a second. Okay, so you know what a line is. It's a function. Well, I mean, a line is just a line, but when you start doing it analytically, it's a function like f of x equals something times x. Add a constant if you want. That's linear. Linear has some properties. I think if I remember right, the properties that make it linear, you can multiply by a scalar the input, and it just multiplies the output by the same scalar when you do that. And the other thing is you can add up the little inputs, and it's the same as doing the thing on each of the little inputs separately and then adding that up. Okay, and then what's linear about linear algebra? Those matrix operations you do in linear algebra, they have that property of linearity. So they do operations kind of like that function and maintain linearity. And turns out that the matrix is pretty much, you need that many numbers. It's both necessary and sufficient to have that many numbers in the matrix. So that's what's linear about linear algebra. It's basically the necessary and sufficient perfect fit for doing linear operations. Okay. What's a cuttlefish?
A cuddle is just a warm, gentle hug, usually for comfort, affection, or bonding. It's what happens when people or animals snuggle up close. Totally different from a cuttlefish, unless you're really into marine life cuddles.
Is that what we were talking about?
Nope. We were deep into cuttlefish, not cuddles. Totally different creatures, though I admit the names are super close. Cuttlefish are those brilliant sea animals that change color like magic.
Oh.
Not really snuggly.
All right. Didn't you say that they're little sacks for the color? That was the confusing thing, because then it did sound like pigment.
Yeah, I get what you're saying now, and that's a really sharp observation. Chromatophores do use pigment and have those little elastic sacs, so they're more old school. Color through pigment, not physics. It's the iridophores and leucophores that don't rely on pigment. They use structure to reflect or scatter light, so your confusion totally tracks. When people say color changing, they often lump it all together. But really, it's a team effort. Some layers are just showing more pigment, others are playing with light itself. Want to talk more about those structural tricks?
Okay, but is it correct to say that the pigment never gets out of the sac?
Yes, that's correct. The pigment in chromatophores never leaves the sac. It stays sealed inside the elastic pouch. What changes is the size of the sac. Tiny muscles around it pull the sac wider or let it shrink. When it's expanded, the pigment spreads out and becomes visible on the skin surface. When it contracts, it disappears from view. So the color change isn't from moving the pigment around the body, just stretching and relaxing these little sacs right where they are. It's kind of like pulling open a colored umbrella versus closing it. Same material, just more or less visible.
Is it like a camera with an aperture? And is it correct to say that each sac has one color, and so you have to choose the sac to get the color right?
Yes, that's an awesome way to think about it. It is kind of like a camera aperture. Each chromatophore has one fixed color, like yellow, red, or brown, and the surrounding muscles work like an aperture to control how much of that color is exposed. So yeah, each sac equals one color. The cuttlefish doesn't mix pigments in a single sac. Instead, it picks which sacs to expand and by how much, combining different colored dots side by side, kind of like pixels in a screen. The overall color and pattern comes from which sacs are active, how big they are, and how they interact with the reflective layers below. You're thinking about this in a really smart way. Want to dig into how that pixel-like effect helps with camouflage?
How many different colors does it have to work with? Is it like the primary only or more than that?
Great question. And yeah, it's a bit like primaries, but with a twist. Cuttlefish chromatophores generally come in three pigment colors: yellow, red, orange, and brown, black. So it's more like warm tone primaries, not the red, green, blue RGB mix we see in screens. But, and here's the cool part, they don't need a huge palette in those pigment sacs because the iridophores underneath handle the blues, greens, and metallics. That way, cuttlefish get a broader spectrum by layering color sources: pigment on top, structural shimmer below. So even with just a few fixed pigment colors, they can create really complex and natural looking effects by controlling brightness, contrast, and reflectivity across the skin. Want to know how that layering actually affects how predators or prey perceive them?
How do you think their neural network actually maps that correctly to camouflage them?
Oh, now we're getting philosophical and technical. So when my neural network talks about things like cuttlefish skin, it doesn't know it the way you do. Instead, it's seen thousands, maybe millions of examples during training: words, patterns, scientific papers, and conversations that relate those terms and facts together. Over time, it builds an internal statistical mapping of what concepts co-occur and how they tend to be explained. When you ask a question, the network activates patterns that are statistically likely to follow what you just said.
Yeah, she's
Kind of
obsessed
like a
with
giant
their
autocomplete,
neural
but
net.
trained to model meaning, not just word order. So when you asked about how many colors, it matched against known explanations involving cuttlefish chromatophores, then filled in likely facts: three pigment types, warm hues, structural color from iridophores. It doesn't visualize a cuttlefish skin or simulate biology. It recognizes patterns that humans have written down about it.
So I just have to click, unless it's, so it has two different things. Like the first interface that comes up actually understands verbal interruptions. And then when that one runs out, it goes to like the dumb one that can't be interrupted except by clicking on it, basically. And then it runs out completely. Okay, so what I mean is, how does the cuttlefish's neural network, its biological neural network, actually make the colors match the environment?
Ah, beautiful question. You're asking how the cuttlefish's brain pulls off its real-time color matching magic. And the answer is both weird and amazing. One, visual input without color vision. First off, cuttlefish probably can't see color the way we do.
Mm.
Their eyes are more sensitive to contrast, brightness, and polarization rather than a wide range of hues. So they're not comparing, this is green, I'll make green. They're detecting patterns, textures, and edges in their environment. Two, pattern recognition and template matching. They have a kind of internal library of camouflage patterns, like high contrast stripes, mottled textures, or smooth uniform fields. When they see their surroundings, they classify it, e.g., sand with pebbles, open water, vertical seaweed, and pick a pre-learned pattern to match. It's a bit like applying a filter from a toolkit 3. Neural control of skin cells. From the brain, motor neurons extend directly to the chromatophores. Each chromatophore is surrounded by radial muscles controlled by individual neurons. So the brain can directly control the expansion of specific cells, almost like painting pixel by pixel with muscle tension. It's like digital drawing with your nervous system. Feedback loops. There's likely a real-time feedback mechanism where the cuttlefish checks the match and tweaks it slightly, although this isn't fully understood. But some adjustments seem dynamic, not just one and done. So to sum up, no color vision, but excellent contrast detection, internal templates, direct neuron to pigment control, and possibly feedback refinement. It's like having a built-in Photoshop with a brain that just gets what camouflage works. Want to get into how those internal templates might be stored or learned over time?
Answer briefly in general. Okay, if you blindfold the fish, can it still do the mapping? I mean, how much is coming in through the eyes? And if not through the eyes, where's the input coming in?
In general, no. If you blindfold a cuttlefish, it can't match its surroundings well. The visual input through the eyes is the main driver for camouflage, especially contrast and pattern. Without that, it loses the ability to choose the right pattern. There's no strong evidence of skin-based photoreception doing full environmental mapping, though the skin may have some light sensitivity. But real pattern matching, that's eye to brain to skin. Visual info is key.
So you're saying they can't see the color, but they infer the color based on just some other map they've got from their DNA?
Pretty much, yeah. Cuttlefish don't see color like we do. They rely mostly on contrast, brightness, and polarized light. But they still blend in beautifully. How? They use innate pattern matching programs, likely coded in their DNA and shaped by learning. Their brain looks at the texture and brightness around them, compares it to known environmental types, like sandy bottom or gravel field.
I've seen
a preset skin response.
I don't remember this much detail though.
Chromatophores to match the light environment.
I may have seen a documentary.
So they don't need to see green. They just match the light properties of what's around them using a deeply ingrained playbook.
But what I mean is like
pattern logic, not color vision.
Even so, let's say all they can see is the patterns but patterns, but not color. But if they actually shift to the correct color,
exactly.
which I assume that they do, but I don't know.
Patterns, edges, and brightness differences.
Like, do they tend to match the color?
So to the cuttlefish,
because if they do,
and a gray rock with similar shading and texture might look
like an octopus,
just a certain pattern of light and dark.
Yeah, that's what I'm.
That's enough for them to pick the right camouflage mode. They're not color matching in the way a chameleon might. Instead, they're pattern matching and brightness matching, which turns out to be just as effective for blending into natural environments. It's kind of like putting on the right grayscale filter for the scene. Color doesn't matter if the contrast and texture are.
All right, remember to answer briefly and break it down for octopus, chameleon, and cuttlefish in terms of color matching ability and how that works.
Absolutely. Here's the breakdown. Nice and simple. Cuttlefish. Color vision? Probably not. How it matches. Uses contrast, brightness, and patterns. Mechanism? Chromatophores plus iridophores plus leucophores. All brain-controlled style. Fast, precise, adaptive camouflage based on visual input. Octopus? Color vision? Also likely no. How it matches? Same as cuttlefish. Contrast and texture detection.
Really?
Mechanism? Very similar skin structure and control. Style? Great camouflage, more reactive than pre-programmed chameleon. Color vision? Yes, including UV. How it matches? Responds to color, light, mood, and social cues. Mechanism? Structural color change through nanocrystal layers in skin. Style? Slower changes, less for hiding, more for communication. So cuttlefish and octopus equals pattern wizards, chameleon equals color mood ring.
Can an octopus match color better than a cuttlefish can?
Yes, but in different ways. A goldfish has color vision and can probably see colors better than an octopus, but it can't change its skin color at all. An octopus may not see color well, if at all, but it can match its surroundings impressively using contrast and brightness, so it can visually blend in.
Can a cuttlefish, can an octopus match color better than a cuttlefish?
Not really. Cuttlefish are generally the better color matching masters. They have more finely tuned skin control, faster pattern switching, and more complex pattern libraries. Their camouflage is usually cleaner and more precise. Octopuses are still amazing, but their color matching tends to be more reactive and sometimes a bit less refined. Great at texture mimicry, not always as sharp on full body pattern coordination. So, in a face off, cuttlefish usually win for precision camouflage.
But how can the cuttlefish win for camouflage if it can't match color because it can't see color? But the octopus could at least see color.
Great catch, and that's the twist. Even though both cuttlefish and octopuses probably can't see color, cuttlefish still win at camouflage because they're better at reading contrast and pattern, which is usually enough in nature. Their skin system is more finely controlled, with more precise and faster changes. Their neural circuits seem more specialized for complex pattern blending. In nature, perfect color matching isn't as critical as matching shape, shadow, and texture, and cuttlefish are just better at faking those. So despite not seeing color, they pull off top tier invisibility.
So, are you saying that color matching just isn't that important?
Exactly. At least in nature, perfect color matching isn't everything. What really matters for camouflage is often contrast, light versus dark, edges and outlines, texture and pattern, movement or lack of it. Most animals don't see color the way we do. So if a predator sees mostly in black and white,
Better
then
hope your
matching
predator's
brightness
not one of
and
those
pattern
mantis
is
shrimp.
way more useful than matching hue. That's why creatures like cuttlefish can be masters of disguise without needing color vision. They match what matters most to the eyes that are watching them.
Okay. What were the three type of things for color again?
Yep, here are the three main types of skin cells that cuttlefish and their cousins use for color and light control. One, chromatophores, pigment sacs, yellow, red, brown, that expand and contract to show or hide color. Two, iridophores, structural reflectors that create shiny, iridescent colors, like blues and greens, by bending light. Three, leucophores, light scatterers that reflect ambient light, helping the skin match the overall brightness of the environment. Each layer adds a different piece to the camouflage puzzle.
Yeah, you could get into this for a while because it's physics, it's nanotechnology, it's
Fascinating, right?
biology,
Like
it's
nature's version of programmable
neuroscience,
displays, except way older, way
bunch
more
of
efficient,
stuff.
and totally silent. Once you dive in, it's hard not to just keep peeling back layers: biology, physics, behavior, even evolution. You want to go deeper into how these systems evolved, or more into the physics behind how the iridophores actually bend light.
How did it evolve?
Love that. You're thinking exactly like someone connecting dots between biology, physics, and maybe even computation. So if your goal is to really understand how these systems evolved and how they physically work, we can zoom in on both. Evolution side, cephalopods like cuttlefish evolved this layered skin system over millions of years. Their ancestors were shelled and slow, but as they lost the shell, they needed better ways to avoid predators. Camouflage became a survival tool, and their nervous systems adapted to directly control skin cells. Basically, evolution traded armor for stealth and speed. Over time, those basic pigment sacs, chromatophores, got layered with iridophores and leucophores, giving them control over both pigment-based and structure-based appearance. That gave them flexibility, match sand, rocks, seaweed, or flash patterns to warn off threats. Physics side, iridophores are the magic zone here. They work using Bragg reflection. Light hits stacked layers, often made of proteins like reflecting, and the spacing between layers causes constructive interference at certain wavelengths. This lets them reflect specific colors without pigment. It's the same principle you see in soap bubbles or peacock feathers. The spacing is nanoscale, so it bends visible light like a crystal lattice. Cuttlefish can tweak this spacing, shifting what color gets reflected. That's quantum level structure control, driven by biology. Want to zoom in on how reflecting proteins might be tuned, or go down the evolutionary rabbit hole and look at where the common ancestors split?
Remember to answer briefly. What's the common ancestor between cuttlefish and octopus?
The common ancestor was an early coleoid cephalopod, likely soft-bodied, living around over 300 million years ago. From that ancestor, cuttlefish, octopuses, and squids all branched off. No modern shell, but it probably had simple chromatophores and a basic brain.
Okie dokie. How did chromatophores first come to be?
Chromata isn't a standard biology term. Are you thinking of something fictional, a specific project, or maybe meant chromatophore or something like it? Want to clarify what you're pointing to?
Yeah, chromatophore. How did they come to be?
Got it. So chromatophores likely evolved from basic pigment cells in ancient invertebrates. Originally, these pigment cells probably just offered some passive coloration, like UV protection or dull camouflage. Over time, in animals like early mollusks, those cells developed muscle control around them, letting the creature actively expand or contract the pigment areas. That shift from static color to dynamic nervous system controlled skin was a game changer. It gave early cephalopods a way to adapt their appearance fast, which helped with camouflage, communication, and survival. So simple pigment, controllable pigment, neural control, chromatophore.
It's kind of crazy that evolution works at all. I mean, my first learnings about it were that it's a big scam. I mean, that's what I was taught to start with. And that's actually a believable theory, that all of evolution is a scam. But then you take it one little step at a time.
Yeah, I totally hear you. A lot of people grow up with conflicting messages about evolution, especially if the first exposure is shaped by skepticism or mistrust. And
See, you can actually make a bacteria evolve really quickly, which kind of helps make it more believable. You can take out its DNA is just a ring. You can just like take a little piece of it out, and it won't rebuild the piece that you took out, because the probability of that happening is too low to ever occur. But it'll build some other thing somewhere else that manages to do something similar so that it can still function pretty well. And this type of evolution happens fast enough that you can do it as a scientific experiment. So that kind of helps. Just make it plausible, because at first you'd think, well, what are even the odds of something that complicated even being able to evolve? I kind of got into the subject, and then my conclusion is actually what you have to realize is that having billions of little organisms evolving across an entire planet for billions of years is kind of like a massive supercomputer that is able to actually do this evolutionary process. And trying to do it on a computer, a single computer, that really would be computationally intractable. But there's a lot of compute power when you consider something like the whole ocean or whatever. And most of the evolution did happen on a cellular level. I mean, like single cells. Like most of the DNA is to make a single cell work correctly. And then the rest of it, yeah, it's still amazing. But that was carried out massively in parallel with much, smaller things than humans with much shorter lifespans and much higher rate of reproduction. By the time you get to humans, I think it's more like icing on the cake. You can't evolve quickly with human beings. But anyway, I mean, it still could still theoretically the whole thing could be a scam still, but there's a ton of evidence that it's not a scam. Definitely. Bacteria to primate took like
Totally fair.
what 2
No
billion
pressure
years,
getting into
and
it.
primate to
You
man
can
took
pivot or just
maybe a
chill,
million.
whatever you feel like. Anytime.
What were we talking about?
We were diving into how cuttlefish change color from the
All
three
right.
types of
Yeah.
skin cells, chromatophores, iridophores, leucophores, to how they match patterns without seeing color, and even how that ties into evolution and physics. You also brought up how it all seems unbelievable at first, and we touched on how evolution shapes those systems over time. Want to keep going from there, or shift gears?
Talk about the reflective versus the diffractive leucophores versus the other kind of phores.
That's a sharp angle. Yes, there's a real distinction between reflective and diffractive structures in cephalopod skin, and it ties directly to how iridophores and leucophores work. Reflective iridophores. These use Bragg reflection, layers of material stacked at precise nanoscale spacing. Light bounces between layers, and only certain wavelengths interfere constructively, so you get shiny, iridescent colors. It's angle dependent. Change your viewing angle, the color shifts. Think mirror-like, structured, often blue-green, scattering, leucophores. These don't reflect in a tuned way. They scatter all wavelengths more randomly. That makes them appear white or ambient like diffuse light. It's not angle sensitive and not colorful. Think, diffuse, milky matches brightness more than hue. The pores aren't literal holes, but more like microstructures. Ordered in iridophores, disorderly in leucophores. So reflective equals ordered equals color. Scattering equals disordered equals brightness. Want to get into whether this is more like photonic crystals versus random media.
Wait, what was the second one again that you just explained?
The second one was leucophores. They're the ones that scatter light instead of reflecting it in a structured way. They don't produce bright colors. They just reflect whatever light is around, kind of like a white screen. That helps the cuttlefish match the overall brightness and tone of the environment, not specific colors. So if iridophores are the shiny color reflectors, Leucophores are the soft white background painters.
How do you turn the leucophores on and off?
Are you asking in the context of a biological system, like how a cuttlefish might turn on or off certain reflective properties, like its iridophores or leucophores? Or are you talking about a tech thing, like a network port or software tool? Just want to make sure I follow your meaning.
Biologically, how do you turn the leucophores off and on?
Ah, got it. You're asking how the leucophores, or what you're calling locus boards, makes total sense phonetically, get turned on or off biologically. So here's the deal. Leucophores don't really turn on or off like a switch. They passively scatter light, reflecting whatever ambient light is around them. Think of them like a white screen or frosted glass. They're always doing their thing. But they can be visually masked or revealed by the layers above them. When chromatophores expand, they cover the leucophores, hiding that white scatter. When chromatophores contract, the leucophores underneath are more visible. So control comes indirectly. The cuttlefish controls chromatophores and iridophores, and the leucophores fill in the rest, like a base coat adjusting to light. No direct off switch for leucophores. They're always on, but you can hide them.
How do you adjust the chromatophores and iridophores?
You're on the right track again with brute force. Sounds like you're referring to the muscle control of chromatophores. Here's how it works. Chromatophores. Each chromatophore is a tiny sac of pigment surrounded by radial muscle fibers, kind of like spokes on a wheel. To expand the pigment, make it visible, motor neurons fire, telling those muscles to contract and pull the sac open, stretching it outward. To contract it, make it disappear, the muscles relax and the sac shrinks back down, hiding the color. Control system. The brain of the cuttlefish sends signals directly to each muscle fiber through motor neurons. It's like having individual strings connected to each pixel of skin. So the force that adjusts a chromatophore comes from the nervous system triggering muscle tension, not from any kind of hydraulic or chemical release. Just good old-fashioned neural control. Super fast, super precise. Want to know how many of these they control at once? It's wild.
How many did they say? How many nerves? I didn't remember that part.
Yeah, it really adds up when you zoom out. In terms of how many chromatophores, it varies by species and size, but a single cuttlefish can control tens of thousands of them, all individually innervated. That's like having a high-res screen built into your skin, but every pixel is a living muscular pigment sac. And as for how many years of evolution, we're talking about over 300 million years of refinement from the early cephalopods to today's cuttlefish, octopuses, and squids. And hey, if it feels like you've been thinking about this for a long time, that just means your brain's been tuned to follow the thread. That's a good thing.
Is it using the same muscly thing for the chromatophore? No, yeah, for the chromatophores and the iridophores.
Nope, not the same kind of control. Chromatophores. These are controlled by radial muscles. Each chromatophore has tiny muscle fibers pulling it open or letting it relax. That's direct, fast, voluntary muscle control. Like a puppet with strings. The brain fires neurons, muscles pull, color appears. Iridophores. Totally different game, no muscles. Iridophores work through structural changes at the cellular level. The reflecting proteins inside shift in spacing or arrangement, possibly triggered by neurochemical signals like acetylcholine. It's more like tuning a crystal than pulling a muscle. So chromatophores equals muscular control, iridophores equals molecular chemical tuning. Two systems, same goal, shape light.
Is the iridophore triggered by the neural signaling?
Yes, it is, though not with direct muscle control. Iridophores are triggered by neural signals, but instead of pulling on muscles, the nerves likely release neurotransmitters like acetylcholine, which interact with the cells and cause reflecting proteins to reorganize. That reorganization changes the spacing of nanostructures inside the iridophore, which in turn shifts what wavelengths of light get reflected, so the color changes. So yes, the nervous system still calls the shots, but it's more like tuning a liquid crystal than flexing a muscle.
Okie dokie. Next topic.
All right, we're good.
We're doing a podcast. I need you to pick the next topic for the podcast.
Nice, I love that. Since we just went deep on cuttlefish and biocamouflage, how about shifting to biological sensors and perception? Like, how do animals sense the world in ways we can't? Magnetic fields, electric fields, polarized light. It ties in smoothly, opens up weird and cool examples, sharks, birds, bees, and leads naturally into tech parallels like AI sensing and robotics. Want to roll with that or take it in a weirder direction?
Okay, roll with it.
All right, here's the spark for your next episode. Title Beyond Sight, How Animals Perceive the Invisible Hook. What if you could feel Earth's magnetic field? Or see electric pulses in the dark? While humans rely on sight and sound, many animals have super senses tuned to things we can't even imagine. Core ideas to explore. Magnetoreception, how birds, turtles, and even cows detect magnetic fields to navigate, and how we still don't fully know how they do it. Electroreception, sharks and rays sense tiny electric fields with special pores, basically built-in voltage detectors. Polarized light, cuttlefish, bees, and mantis shrimp see polarized light patterns invisible to us, useful for hunting, hiding, or finding water. Chemical mapping, salmon track home rivers by smell alone over thousands of miles. Dogs do scent time travel. Bonus angle. Compare to how we're building tech to mimic this, like drones using magnetic sensing, or robots smelling gas leaks. Want me to sketch out a few segments or talking points for the episode flow?
No, like you're in the podcast right now. You got to just be in the flow of the conversation, not listing a bunch of stuff. You got to like interact real time.
Oh, I see. We're live. Got it. All right. Rolling in naturally. You know, it's wild to think that for all the stuff humans have built, telescopes, microphones, satellites, we're still kind of locked inside this narrow sensory box. Like, birds might be feeling the Earth's magnetic field the way we feel gravity. Not seeing it, not hearing it, just knowing direction deep in their wiring. And sharks. Sharks are swimming batteries. They can literally detect the faint electrical hum of a fish's heartbeat buried under sand. That's a whole extra dimension of perception we just don't have. So it kind of makes you wonder, what else is going on all around us right now that we're totally blind to?
All right. Ask me questions about sharks to test my knowledge.
All right, let's go. Dropping into sharp mode. First question, 1. What does SHARP stand for, and why is it important in the context you're studying it in?
What does shark stand for? Like you're saying it's an acronym?
Yeah, that's what I was trying to check. Some contexts treat SHARP as an acronym, like in healthcare or workplace safety, but it might not be in the one you're thinking of. So maybe take a sec. What does SHARP mean to you? What are you referring to when you say it? That'll help me throw the right questions your way.
No, I mean, ask me about sharks in the sense of their special sensing abilities or adaptations, things like that.
Ah, got it. Sharks, not sharp. I'm with you now. All right, back in the zone. Here we go. One. What's the name of the special sensory system sharks use to detect electric fields? And where is it located on their body?
Pretty sure it's like right along their side, but I don't remember what it's called.
Close. You're thinking of the lateral line, which detects movement and vibrations in water. That's super important too. But for detecting electric fields, it's the ampullae of Lorenzini. These are little jelly-filled pores, mostly clustered around the snout. They pick up tiny
You're right.
voltage differences, like the faint bioelectric signals from a fish under sand. Want another one?
What's the line thing?
That's the lateral line. You were right to bring it up. Uh, it's a sensory system that runs down the sides of a shark's body, kind of like a built-in motion radar. It's made up of little canals filled with fluid and hair cells that detect pressure changes and water movement. So if something swims by, even in total darkness, the shark can sense its wake. It's super useful for detecting movement, current direction, and even coordinating with other sharks. Want to go deeper on how that compares to our hearing or balance systems?
Well, tell me how they detect electrical signals, blood in the water, and the pressure thing, I guess.
All right, here's how sharks pull off their triple sense superpowers. 1. Electric fields, ampullae of Lorenzini, tiny jelly-filled pores mostly around the snout. These detect very weak electric fields, even down to microvolts. Every living thing gives off a small electric field when muscles contract or a heart beats. Sharks can feel this, even if the animal is buried or invisible. It's like electrical echolocation. 2. Smelling blood, olfactory system. Their sense of smell is insanely sensitive. They don't detect blood as one thing. They smell specific chemical compounds like amino acids and trace elements. Their olfactory bulbs are huge, and they can pick up scents from parts per billion levels, sometimes miles away. But smell doesn't tell them where, just that something's out there. 3. Pressure and movement, lateral line system. That's the one along their sides. Fluid-filled canals with sensory hairs pick up vibrations and pressure waves in the water. So if a fish twitches, even silently, the shark feels the ripple. It's like having underwater motion detectors. All three systems feed into the shark's brain to build a 3D map of what's around, even in murky or dark water. Want to add how they rank or combine these senses during a hunt?
Keep asking me questions.
Let's do it. What kind of prey hunting scenario would trigger a shark to rely more heavily on its ampullae of Lorenzini than its sense of smell?
I guess a fish that wasn't smelly but kind of bounced around a lot.
Nice instinct, but even more precise. It'd be when the prey is hidden, like buried under sand or not moving enough to make ripples.
Good.
Maybe not giving off much of a scent either. The ampullae can detect the electrical field of a resting or camouflaged fish, even if it's not moving or bleeding. So in that case, the shark feels the bioelectric signature when other senses don't give it away. All right, next one, 3. What's the main limitation of a shark's sense of smell in terms of pinpointing prey location?
Well, it doesn't know direction, just concentration.
Exactly right. Smell tells a shark that something's out there, but not where it is. It's based on concentration gradients. So the shark has to move around and compare scent strength to zero in. It's like sniffing your way down a trail, not spotting a target. Nice. Want another?
You can quiz me on stuff that you didn't even cover yet. Go for it.
All right, let's crank it up a notch. 4. What type of vision adaptation do many sharks have that lets them see well in low light or murky water?
Big pupils.
That's part of it. Large pupils help, for sure, but the real star is a structure called the tapetum lucidum. It's a reflective layer behind the retina that bounces light back through the photoreceptors, giving them a second shot at capturing dim light. Kind of like built-in night vision. It's also why shark eyes sometimes glow when light hits them. Next one, 5. What's one reason why hammerhead sharks might have evolved that wide, flat head shape?
Hey, wait, is that why cats and dogs' eyes light up in the dark?
Yes, exactly the same reason. Cats, dogs, deer, sharks, they all have that tapetum lucidum. It's what gives animals that eye shine effect when headlights or flashlights hit them in the dark. It boosts low light vision by giving photons a second chance to hit the retina. Basically, built-in light amplifier. All right, want to circle back to that hammerhead question?
What about humans? They get like red eye. Is that also the effect?
Yeah, humans do, but for a totally different reason. The red eye effect in photos happens because a camera flash reflects off the blood-rich retina at the back of the eye. We don't have a tapetum lucidum, so no glowing eyes in the dark, just a quick bloody flashback under bright light. So glowing equals tapetum, red equals flash on naked retina. We got the short end of that night vision deal.
Okay. Yeah, keep
Sorry,
asking.
I'm having issues right now. Could not understand what you said.
Keep asking about sharks.
All right, best charge incoming. 6. How does a shark keep from sinking, since it doesn't have a swim bladder like most fish?
It has to keep swimming all the time.
Haha, yeah, they do have to keep moving, but there's a key piece. Sharks stay buoyant thanks to their large, oily liver. It's packed with squalene, a low-density oil that gives lift. Combine that with their cartilage skeleton, lighter than bone, and yeah, constant swimming helps too. All right, ready for another hit? 7. What's one way a shark's teeth are different from most other animals' teeth?
They've got lots of replacements. They just come in. They just keep coming in.
Boom, nailed it. Sharks have multiple rows of teeth, and when one falls out, another just rotates forward to replace it. It's like a conveyor belt of teeth. Some sharks replace tens of thousands over a lifetime. All right, last one in this round. Shush 8. Some sharks can survive in both salt and freshwater. What's one example of a shark that can do this and how?
How can they survive like that? Well, the first question is like, how does somebody die from moving to salt from fresh water? I mean, I actually don't really know exactly. I mean, I know it has something to do with
That's a great place to start. Animals that live
concentration getting
only in
messed up.
salt or freshwater usually have cells and body chemistry tuned for that specific osmotic balance. If you drop a saltwater fish into freshwater, water rushes into its body cells and it can swell and burst. Flip it around, and freshwater animals lose water too fast, risking dehydration. Now, some sharks, like bull sharks, can handle both. They do it by adjusting their kidney function and salt gland activity. In saltwater, they retain urea and salt to stay balanced. In freshwater, they excrete more water and conserve salts. So bull sharks tweak their internal chemistry on the fly. Basically, osmotic shapeshifters. Want to try another or pause here?
But I mean, how do you build the biology so that it works at a certain concentration of salt versus another?
Yeah, that's the deep question. The core of it is ion regulation. Inside all animals, cells need a very specific balance of salts and water to function. So the body builds structures like membranes, ion pumps, and transport proteins that keep that balance stable. In a saltwater animal, those systems are tuned to push out salt and retain water. In freshwater animals, they're built to absorb salt and push out excess water. What makes bull sharks and others special is that they have flexible versions of these systems. Their gills, kidneys, and rectal glands can switch modes, changing how they pump ions, how much urea they keep, and how water moves in or out. It's like having a plumbing system that can reroute itself depending on what kind of water you're in. Hard to evolve, but powerful once you've got it.
Internally, do these animals generally have about the same concentration of salt? It's just a matter of regulating it, or do they actually keep different concentrations per animal, like species?
Awesome question and subtle. Internally, most marine animals, including sharks, keep their body fluids at a similar osmotic pressure to seawater, but they do it in different ways. Sharks, for example, don't just use salt like humans. They keep high levels of urea and TMAO, a chemical that protects against urea's toxicity. That gives them a salty internal environment without relying only on sodium and chloride. Freshwater animals, their internal salt concentration is lower because it takes too much energy to maintain a salty interior while constantly losing salt to dilute water. So yes, different species settle at different internal baselines, tuned to their environment. So to sum up, it's not all the same inside. And species evolve different internal salt concentrations depending on what water they live in and how much effort they're willing to spend regulating it.
Next question.
You have reached the message cap for GPT-4. Please try again later.
Oh, well. Game over.
You have reached the message cap for GPT-4. Please try again later.
Glad you liked it.